our lecture is on cholesterol synthesis and regulation. As you all know, cholesterol is a very, very important constituent of the cells, if you have heard about it before. And this basically comprises a lot to the basically the function and the structure of the cell membrane. So we will be discussing specific learning outcomes inclusive of the chemistry and function of cholesterol, then followed by the precursors and sites of cholesterol synthesis. Then there are steps, enzymes, coenzymes, and regulation involved in cholesterol synthesis. And the last not least, state the action of cholesterol lowering drugs in the cholesterol metabolism. So from different diseases, when the cholesterol is very high, like we got it hypercholesterolemia and many of the diseases which are more towards cardiac issues, they are this topic become very specific and very important. But looking at the learning outcomes, we will be not moving towards that direction in this lecture. It is more to the basic chemistry, function, metabolism, cholesterol for now. But on the other lectures, we'll cover, inshallah, the other parts which are related to cholesterol. So what is cholesterol? Basically, cholesterol is derived from a Greek word "chol," steros, and all three words. So which actually is bile, solid, and alcohol. Because remember, the cholesterol is the precursor of bile acids, and for that reason, it has been given the name. Almost all nucleated cells can synthesize cholesterol. It is widely distributed in the body. So in a 70 kg man, a total of about 140 gram of cholesterol is available, which is basically distributed as about 30 gram in brain, 30 gram in muscles, 30 gram in adipose tissue, 20 gram in skin, 10 gram in blood, 10 gram in liver and spleen, 5 gram in bone marrow, 3 gram in alimentary tract, and 2 gram in adrenal gland. So this is the generalized in the 70 kg man. Cholesterol is a light yellow crystalline solid, and under the microscope, it basically has a notch appearance. So it is soluble in chloroform and other fat solvents. So now the interesting part is this cholesterol is absent in prokaryotes. In plants, cholesterol is absent, but other plant sterols are present. In bacterial plants, compounds similar to sterol exist, and we call them haploids. Haploids. It's very important, but in this lecture, we are not going to discuss the haploids. Cholesterol was isolated in 1758. It's characterized in 1818, and the person who basically initiated the cholesterol structure was given a Nobel Prize in 1927. It is the most important animal steroid from which other steroid compounds are formed. So it is distributed in the animal tissues. Okay, now we go to the chemistry of cholesterol. All steroids have a cyclopentano per hydro phenanthrene ring basically. So this is how the ring is. We got two rings, which is 6 carbon. Then we have a third one is again a 6 carbon, which is remember ABCD rings. Okay, so ABCD ring is basically cyclopentano per hydrophenanthrene ring system. So it is fused ring system made up of 3 cyclohexane rings designated as A, B, and C, and a cyclopentane ring D. The 6 member rings are in phenanthrene arrangement. So this arrangement is called phenanthrene arrangement. The total of 27 carbon atoms, one hydroxyl group at third carbon, which is characteristic of all sterols. So if you see here, we have basically the third carbon here. Wait. So we have the third carbon here. I'm unable to use the function for annotation. I don't know what is the reason for that. Okay, so we have the third carbon here, which is having the hydroxyl group, and then what we got here is we have double bond between carbon number 5 and 6. Here we go. So we have a double bond here, and 8 carbon beta oriented side chain attached to the 17th carbon. So here we got the 17th carbon, and with that we got total 8 carbon chain: 1, 2, 3, 4, 5, 6, 7, and 8. This is a basic structure for arrangement of basically fen and therine arrangement all together. So this is how the structure of cholesterol is, and a little bit of its basic biochemistry, basically the biochemistry is a chemistry. And then functional cholesterol, very important, because number one is it is involved in basically what we call the integrity of the cell membrane. Cholesterol is a component of cell membranes and has a modulating effect on the fluid state of the membrane. Remember the inside the cell membrane, we have the lipid bilayers, we have all those different kind of lipids. So one of them is the cholesterol. More cholesterol means a different thing. Less cholesterol mean in the cell membrane a different thing. The function of the cell membrane will totally be changed. We'll discuss a little bit about it in the next slides, but I'll give you the material, but you have to read it yourself. I'll not cover everything regarding the cell fluidity in this lecture. So number two is the nerve conduction. Cholesterol is used to insulate nerve fibers. Thirdly, it is involved in synthesis of bile acid and bile salts. Bile salts are important for fat absorption, basically, as you know, and I think you're going to study it more, inshallah, in coming few lectures. Number four is for the synthesis of steroid hormones, which could be glucocorticoids, androgens, estrogen are from cholesterol. Number five is where vitamin D3 is from 7-dehydrocholesterol, which is one of the precursor of the cholesterol. So number six is esterification, the OH group of cholesterol, which we just discussed just now at carbon position 3, is esterified to fatty acid to form cholesterol esters. This esterification occur in the body by transfer of the PUFA multi by lecithin cholesterol acide transferase. Don't get worried if, for example, in your exam you get a question, what is the function of cholesterol? You can write it this way if you want to. But what some of the biomolecules like lecithins, cholesterol acide transferase, PUFA, all these things we're going to cover in the lectures in few seconds. So you can have a more overlook, how it is actually working out. So, are you all okay? Can you all understand me, or I'm very fast?
Yes, Prof.
Okay. So now we move on to the cholesterol source and requirement. So now we could have the cholesterol. Basically, the whole idea is the whole cholesterol will goes basically or enter into the liver cholesterol pool. Inside the liver, all the other functions will be associated with the cholesterols, and then from there it will move on. But the sources of cholesterol which are used to get the cholesterol pool inside the liver could be endogenous and could be exogenous. In the endogenous, we have 500 to 700 milligram per day, and it is synthesized in all nucleated cells of the body. For the exogenous source, it is normally we're taking in our average diet is 300 to 500 milligram per day. So food of animal origin such as meat, liver, brain, egg, yolk, lard, all these things, they got basically the cholesterol which can cover your basic needs. Okay, now about absorption cholesterol, you're taking the cholesterol in your diet, and what happened? The cholesterol ester present in the diet is hydrolyzed by cholesterol esterase. The free cholesterol is incorporated into the bile salt, micelles, and absorbed into the mucosal cell. You remember you see this term you have heard many times. The micelles is basically the first time when they were proposed, it was called hemicells. Basically, half of them is hydrophilic, one half is hydrophobic for the reason. So absorption need micellar formation. There is a specific protein which facilitates the transport of cholesterol into the mucosal cells from the micelles. Inside the mucosal cell, cholesterol is re-esterified and incorporated into the chylomicrons. I'm basically, frankly speaking, I'm trying to just read around so that you can get the basic idea. Where I need is really important to make you understand it. I'll just go back to the animation again, so it will be good for all of you. So the dietary cholesterol reach the liver through chylomicrons. So make it very easy. You got the cholesterol in the diet in the form of cholesterol esters, which is hydrolyzed by cholesterol esterase. Then it goes into the intestine. Inside the intestine, it got absorbed in the form of micelles. When it got micelles inside the mucosal cell, it is re-esterified and it incorporates into the chylomicrons. And the chylomicrons goes into the lymphatic. Okay, from the lymphatic, it goes into the bloodstream. From the bloodstream, it goes into the liver. Is it clear? Very straightforward if you have not read it before. So again, we got the cholesterol esters, through the cholesterol esterase. These cholesterol esterase will be taken up in the intestine in the form of micelles. These micelles will go into the lumen inside the mucosal cell. They will be changed into chylomicrons. These chylomicrons will be taken up by the lymphatics, bloodstream, and liver. So this is how the whole system will work. So plant sterols, for example, cytosterol decrease absorption cholesterol. People who have been taking the plant sterols in their diet, this interact with your basically the animal sterol, which will be taken in the diet, and it will stop the absorption of animal sterol. So what is happening here? You got a protein designated NEC1L1, which we'll call NAMN-PIC-C1 like one protein, which is involved in the absorption of cholesterol. So what actually is happening here? If you can see the picture down there I've shown here. So what actually is happening here? Okay. So what is happening here? The cholesterol esters are taken up at this level by this protein. So when the cholesterol goes inside the enterocyte, inside the intestine, what will happen? Another protein which is called ABC protein, which is ATP binding cassette proteins, and it's a transporter protein, and it also works in the presence of ABCG5 and ABCG8 proteins. So what happened altogether? These three proteins basically control the quantity of the cholesterol which has to be absorbed. But the first protein is involved in the transport of the cholesterol. So that is the first one is involved in the transport. Second one is involved in the quantity. So remember this part. So if, for example, the cholesterol, the amount is too high, they will be excreted or they will be secreted back into the lumen by ABC system, basically. So this is how the absorption of the cholesterol work. So this whole part maybe we know before, but this part could be new for all of us. So remember these two proteins, the first one is the transport protein, the second one is the ATP binding cassette protein, which is involved in the quantity of the cholesterol which is being taken up by the by your cells. So just to add in a little bit of new knowledge to you, because now you are big babies, you know, you have to go for the treating the patients now. Okay, well, now this is about the cholesterol and membrane fluidity, which we have been discussing. I try to make it, don't get worried. These are just the pictures. Relax yourself. It's not, biochemistry is more like mathematics. If you take it like a biology, you'll become complex. It's not, don't take it like a pharmacology and other subject. It's very easy. So what you have to do here, if you can see here, you got, if you can see here, what you got here is basically, this is the basic structure of cell membrane. So if we talk only about the fats or the lipid molecules inside the cell membrane, you go, remember we said it's a lipid bilayer. We got a polar up there, we got a polar down there, and in the middle is a apolar molecules. So now we have all these A, B, C, D, E, F, G, and H. So these are all different kind of lipids which are basically present inside the lipid bilayer of cell membrane, basically. And one of them is, if you look at the G part, this is the cholesterol, basically. So that is how the cholesterol is coming in. Now, the tricky part is here. Remember, the cholesterol is basically involved because it is inside because it's inside these cells, basically. So what actually is happening inside the cell membrane, actually, so the presence of cholesterol basically it regulates the packing of the hydrocarbon tails of phospholipid molecules. More cholesterol means, try to understand, more cholesterol means less fats will go through the cell membrane. So it will provide the integrity to the cell membrane. So it will not, the cell membrane will not feel like a fluid. It will become a little bit rigid now after that. So this character will change many things in the cell membrane, you know, the function, what are the molecules which can pass, which can go in. So this slide is not important from your examination point of view, but to just make you ensure one of the functions of the cholesterol is the membrane fluidity. This membrane fluidity depends on two things. Number one is the temperature. and number two is the composition of this cell membrane. Number one, the temperature, and number two is the composition of the cell membrane. Inside the composition part, cholesterol counts a lot. And when the cholesterol come in, remember, it also have a very serious issue with that temperature. So the more the cholesterol, the lower will the transition temperature through which the things can pass through the cell membrane. So making it easy, remember there is a term called membrane fluidity, and cholesterol is involved in membrane fluidity. That is all what I want you to remember now. Forget all everything, and you don't need it for the examination point, but you must know for the pharmacological applications in the future, you must be needing this word membrane fluidity. That is the knowledge term you need to know. Okay, so we move on from here. Now we said that the cholesterol is basically being absorbed into our into our lumen. So inside the small intestine, what is actually happening? These all cholesterol esters, they come together and they have a chylomicron assembly. They assemble together. Once they assemble together, so what happened with the action of ACAT? What happened? We got a lot of cholesterol esters are formed. So these cholesterol esters basically, what actually happened with these cholesterol esters, they start packaging themselves. They start making small packs. So how these packs are formed? Inside will be the cholesterol ester, and outside will be the protein. Sometimes there will be different combination of proteins and cholesterol ester. So what it means? This cholesterol plus protein, when they are together, this is called lipoprotein. The term is lipoprotein. So you'll be discussing, I think, in your next lecture, but you must know from where they're coming in. So cholesterol plus protein, but there's some other, no, it's not only cholesterol. There's some other lipids inside these lipoproteins. Just to have a look, if you see here in this slide, in the picture on the left, if you can see easily, so if you see the picture on the left, you can have the difference between this is how the packaging happened. You got the esters inside and you got the protein outside. So these are different kind of, if you compare this part with all different kinds of, we have LDL, we have HDL, we have VLDL, we have IDL, we have the chylomicrons. So if you look at them, you can easily now understand what is the composition. For example, we talk about LDL now. So inside LDL, we have the apoprotein, we have free cholesterol. Here's the free cholesterol. We got the phospholipids, we got cholesterol ester, and we got triacylglycerols. So this is the composition of LDL, and the carrying protein is B100. Similarly, VLDL, this is the entire composition. Down there, this is the protein which is helpful in their transport. So they become like a kind of particles which will be transporting into the whole body, and then we have their density, everything is down there. But it's not a part of my lecture, inshallah, but maybe it will be useful for you in the future. So what is happening? These different proteins, they move around in the whole body, taking up this cholesterol in the different form and different, constituents inside the cells, outside the cells, towards the liver, away from the liver, into the peripheral tissue. Everything is happening that way. So again, we just summarize quickly. We got the chylomicrons. These chylomicrons are coming from the cholesterol esters, which actually were absorbed in the form of micelles, and these micelles actually went into the in the form of chylomicrons. They went to the lymphatics. From the lymphatics, they go to the bloodstream. From the bloodstream, they came into the liver. So inside the liver, all the other excretion, whatever other reactions happen, and from there it is distributed into the whole body, or it is taken up back into the liver pool. So that is whole stuff which is actually needed for you for now. So now we move on. A very important concept. I need all of you to be highly focused on this slide, because this slide, if you will understand it right now, it will be very easy for you. Will never forget. So easiest part is we go to the step number one, make it very easy. Remember, we have the acetyl CoA. This acetyl CoA is coming, maybe example from citric acid cycle, with the most integrated point for all the pathways is acetyl CoA. Remember, from acetyl CoA we enter into the citric acid cycle. From there we go to the lactone transport chain, and from there we go to ATP adenine. So you focus on now acetyl CoA. Can you all focus on acetyl CoA? I need to show some pictures. before carboxylic cycle, tricarboxylic acid. Okay, so we focus on acetyl-CoA now. So two molecules of acetyl-CoA basically they basically combine in the with the form of acetyl-CoA synthetase, and they are basically what we call it thiolase, and they give us acetyl-CoA. Remember, two acetyl-CoA joined together to give acetyl-CoA. So this acetyl-CoA basically is further one more molecule of acetyl-CoA join this acetyl-CoA. So now in tiga acetyl-CoA, three molecules of acetyl-CoA we are starting up with. This is one, this is two, and the third one is coming in here. So we have now three molecules of acetyl-CoA. Every reaction COSH is released. First one and second one here, it is released when acetyl-CoA will be joining here. And the enzyme here is HMG-CoA synthase. So the next molecule which will be synthesized after three molecules of acetyl-CoA will be fused in the previous two reaction. This is called this is called 3-hydroxy 3-methyl butyryl CoA. 3-hydroxy 3-methyl butyryl CoA, but many of the people never use this term except in pharmacology now. So for as a medical doctors, at least you must know the term HMG-CoA. Remember this term HMG-CoA for now. Okay, so this HMG-CoA it is reduced by HMG-CoA reductase in the presence of NADPH. NADPH should be converting to NADP. So the coenzyme here is NADPH. It will be converted to NADP, and again the COSH will be released here. And then what will happen? The next molecule will be mevalonate. Mevalonate. So remember this name, mevalonate. So very easy. First name was acetyl-CoA, second name was HMG-CoA, third one is mevalonate. So this mevalonate will go through few steps, and we got isopentyl diphosphate. This isopentyl diphosphate is basically C5. It's a 5-carbon molecule. Okay, remember we come back, we go up there, the molecules and everything, the whole the reaction is there. So this is a C5 it's a C5 5-carbon molecule, and then goes through a total of 21 steps in which lot of isoprene units are added, and we got a 30-carbon molecule which is called squalene. So remember again, you have to remember by heart basically 3-acetyl-CoA, HMG-CoA, mevalonate, isopentenyl diphosphate, squalene, and then from squalene we got C27, which is dihydrocholesterol. So here we go, we got the dihydrocholesterol, and remember this is one of the precursor of vitamin D synthesis. Here it can also enter towards vitamin D. Actually, from this level, vitamin D synthesis can come in, and from there you got cholesterol, which is again carbon 27. So this is easy enough for you to go through, which is needed for the medicine purpose for now, and you can it will be helpful for your clinical years. So now this is how the structure of cholesterol is. We have already discussed about it. So the primary site is liver, synthesis is liver. Secondary site involved adrenal cortex, ovaries, testes, placenta, and skin. Cellular compartment involved is cytosol and endoplasmic reticulum. All carbons are from acetyl-CoA. Remember, we said the precursor is acetyl-CoA, and it required NADPH, ATP, and oxygen for sure. So this is how the cholesterol biosynthesis occur. I'll try to make it very simple for the steps which are needed for you as a medical student. So please remember at least these steps. Don't go for so complex like in Harper and the books. Make it very simple what is actually needed for you for now. Okay, now we have the biosynthetic cholesterol, the same thing we are explaining, which is mentioned now in the books. Let us go back what the book says. They said the step number 1 is the condensation, in which the acetyl-CoA they combine together. Second one is the production of HMG-CoA. I will not read down there because you already know about it. The first step is the condensation. We have just discussed about it, in which we need the acetyl-CoA synthesis, and which we call thiolase. Then we have the production of HMG-CoA. Third step is the committer step, in which HMG-CoA reductase is involved. and it is in the microsomal, which is endoplasmic reticulum enzyme. So it used two molecules. It's the NADPH step. Remember we said, and remember, because NADPH is what, I mean, HMG co-reductase, we called it a rate limiting step. Okay, by definition, rate limiting step mean anything which can also be, remember, anywhere where is the word rate limiting step come in your study, it means this step can be involved for the therapeutics, for the medicine, for the any kind of disease prevention, disease treatment. So normally we use the word rate limiting step for all those kind of phenomena, not only for the chemistry of the reaction. So it's a rate limiting step, means it's very important for the medicinal point of view, for the treatment, for the therapeutic point of view. Remember this thing from here on, the rate limiting step. So now then we have the malonate, and then we got basically from the malonate we move on to the next step, which is isopentenyl pyrophosphate. And then we have the condensation of 5 carbons, in which we got the 30 carbon molecule, which is squalene. We have just mentioned in our previous slide. And this squalene goes through cyclization, and then we call lanosterol, but we have to not go into that direction. It's too complex for you. I've actually cut down this part. We have the cyclization, and the cyclization, we cutting to size is there, in which the thymosterol is formed, and the desmosterol is formed, and finally the cholesterol is formed. So these are different, but at least you must know these terms. What is thymosterol is there, desmosterol, these are all different terms. But what you need for your for as a medical student, this is what actually you are into. These are all the reactions which are very important for you. Okay, now we move on to the regulation of cholesterol synthesis. Remember our previous theory, always we talk about the genes and proteins. So we have the genes, we have the transcription, we have the translation, and then what is happening? We are talking about the gene which is involved in the involved in the formation of enzyme HMG-CoA reductase. So if example we stop basically the expression of the gene which is involved in the HMG-CoA reductase, means you have the lesser HMG-CoA reductase, it will be lesser cholesterol synthesis. So we remember the allosteric mechanism, means the substrate is high, the reaction becomes slow. That's the reaction, the substrate is low, the reaction becomes high. Similarly here, when the cholesterol in the cells is very high, it goes and it blocks the transcription of HMG-CoA reductase basically. And when an HMG-CoA reductase is slowed down, then HMG-CoA will be low and cholesterol will be low. Cholesterol amount will go, quantity will go down. But when the cholesterol quantity is less inside the cells, then what will happen? Then the gene and transcription and translation of HMG-CoA gene will increase, and then the cholesterol synthesis will increase. So this is the first step, which is substrate-based regulation, or allosteric regulation could be applicable here. So the other things in this slide, the red one, if you see here, there are the red molecules. Example, not a red, for the girls it will be a very big issue. It's a pink actually. So the cholesterol, the inhibitors are shown in pink, and the activators are shown in brown. So the HMG-CoA reductase is basically also activated by insulin and thyroxine, and it is inhibited by glucagon, cortisone, and statins. Statin is one of the medicine which is taken basically for to lower the cholesterol, especially when you are getting to the higher risk. We'll discuss about it, what it means by the higher risk. So regulation and transcription, we'll just go through and read it in few seconds. The regulatory enzyme is HMG-CoA reductase. Remember, we said we called it's a rate limiting step. The rate limiting step is coming in, we are controlling it. The regulatory enzyme is HMG-CoA reductase. Long-term regulation involves regulation transcription of the gene for HMG-CoA reductase. When sufficient cholesterol is present in the cell, transcription of the gene for HMG-CoA reductase is suppressed, and cellular synthesis of the cholesterol is decreased. When cholesterol in diet is low, synthesis is increased. Okay, so it's very straightforward. Okay, now we move on to the second one, which is regulation of cholesterol but through another molecule which is called sterol regulatory element. So it's inside the DNA and inside in two genes basically. One is HMG-CoA reductase gene, and second one is LDLR, which is LDL receptor gene. So these two genes basically they have the elements. Remember our first concept last year we had the lectures in which we did talk about. I think you were not there. I teach to the other class. Did I teach you about the regulatory elements inside the gene? Yes or no? Okay, inside the gene or inside the promoter of the gene, there are the regulatory elements. So these are element, what actually element is? Element is a sequence of the DNA. It's a DNA sequence to which specific protein can bind. So specific protein bind on the DNA at very specific positions. For example, if the sequence of the DNA is example A, T, G, C, C, T, only specific protein will bind. But if the sequence change to A, T, T, G, C, another protein will bind. So all these proteins, they actually recognize a specific sequence on the DNA where they come and bind. So this sequence of DNA is called the element. So what we are saying here that these two genes, HMG-CoA reductase and LDLR, they are having a specific regulatory element which is called SRE. It's called sterol regulatory element, which is in their DNA. So SRE binding by sterol regulatory element binding protein. So to this SRE, that is the sterol regulatory element, there is a protein which is called sterol regulatory element binding protein, SREBP. It binds for the transcription of these genes. Which genes? HMG-CoA reductase and LDLR. So what is happening when cholesterol levels are sufficiently high? The SREBP remains as an inactive precursor. It binds and activates an SREB cleavage activation protein, which is in intracellular cholesterol sensor. So make it very easy. If I'm sure you get a little bit confused, make it very easy. Remember, there is a DNA sequence which is called sterol regulatory element. And to this regulatory element, there is a protein which bind, which is called sterol regulatory element binding protein. Number one is the DNA sequence, which is called sterol regulatory element or SRE. To this sequence, a protein bind, which is called SREBP, that is sterol regulatory element binding protein. And then the transcription of these genes occur. So what happened when the cholesterol is very high? There is another protein which is called SREBP, cleavage activation protein, cleavage activator protein. So it comes in basically. So what happened? It is a sensor for the cholesterol basically. So when cholesterol is less, this cleavage activator protein, which is SCAP, what happened? It scored SREBP to Golgi bodies. To do Golgi apparatus, proteases are involved, which actually help in the activation of SREBP by cleavage, and then it can help in the transcription of these genes. Are you clear? Are you understanding? I'm trying to make it very simple.
So, Prof, basically when the level of cholesterol decrease, so the SREBP will be escorted by the scab to the Golgi, and the Golgi will cleave it to activate the transmission of
Exactly.
HMG-CoA.
Alhamdulillah. Yes, Afiq. Yes. So
Okay.
inside the Golgi bodies, then these two SIP and SIP2 basically proteases, they will act and they will cleave it, and then all the transcription will start. Okay, thank you. So Afiq, if Afiq understand, I think all of you understand, yes? Or you want me to repeat it? Okay, but in case you need any help, please come back to me again. So number 3 is the covalent modification, which is basically short-term regulation by covalent modification of the enzyme. AMP activated kinase phosphorylates the enzyme, which is inactive. Dephosphorylation by protein phosphate 1 makes it active. So insulin activates the protein phosphatase, whereas cyclic AMP activated inhibitor prevent the dephosphorylation of the enzyme. So you make it very easy. Remember the picture on the right side, the figure on the right side. Remember the inhibitors are in pink, and we have the insulin, how they're working, everything is there. So make it very easy. We have the covalent modification, which is basically phosphorylation and dephosphorylation, in which AMP and cyclic AMP will be involved. This is the third mechanism. And then followed by the fourth one is the insulin and thyroxine increase the activity of HMG CoA reductase. Number five is cortisol and glucagon decrease its activity. Number six, drugs inclusive of lovastatin and other statin group of drugs are competitive inhibitors of HMG CoA reductase. So they are used in clinical practice to reduce cholesterol level in the blood. Okay, so this is these six regulatory mechanism. Number six, I take it under the drugs, but it will not normally it's not covered in the mechanisms and regulations. Tapi you can still use it if you have to, because still they are regulating the mechanism by interacting with all the above five mechanisms. The drugs normally work. Most probably the first one is most important for the drug interaction. Okay, now we move on to the next part, cholesterol pool and the cholesterol metabolism. The total body cholesterol content varies from 130 to 150 gram. LDL, which is low density protein, transport cholesterol from the liver to the peripheral tissue, and HDL transport cholesterol from the tissues to the liver. Actually, I requested, but what to do, because this lecture should come after the lipoproteins. So, but okay, we are still okay. It's still very good for all of you to know it before lipoproteins actually. So total, we have the LDL now, we have the HDL. LDL is involved in transport of cholesterol from the liver to the peripheral tissue. HDL is involved from the transport of cholesterol from the tissue to the liver. So cells of extrahepatic tissue take up cholesterol from LDL. The free cholesterol release within the cell has the following fates. Number one, incorporated into the cell membrane, as we discussed one of the functions. Number two, metabolized to steroid hormones, especially in adrenal cortex and gonads. Number three, esterified with saturated fatty acid and stored in the cell. The enzyme acyl cholesterol acyl transferase help in this reaction. So remember, when we go back, the people who have been taking the lipid lowering drugs, example statins, they are asked to request to decrease their free fatty acid content and their fat contents in the diet. The reason is number three, because if they're still taking the statins, they're still taking the fatty acids, saturated fatty acid in their diet, what will happen? These fatty acids still go and they will be what you call they will be storing inside your cells. Number four is defined with polyunsaturated fatty acid, that is PUFA, by the action of lecithin cholesterol acid transferred and finally excreted through liver. So if you increase, if you now you have to just a small assignment, I have no marks, but for you to read only, read what will happen if you take in diet the PUFA based oil and non-PUFA based oil in your diet. What will happen to your cholesterol synthesis, metabolism, and also the fat metabolism? Just very important for you as a medical doctors. Number 4 point, and it will also affect number 3 point. But read about number 4. Okay, we talk about a little about the excretion of cholesterol. Average diet contains about 300 milligram of cholesterol per day. Body synthesizes about 700 milligram of cholesterol per day. Out of this 1000 milligram, about 500 milligram cholesterol is excreted through bile. So it is excreted through bile. This cholesterol is partly reabsorbed from the intestine. Vegetable contain plant sterol, which inhibit the reabsorption of cholesterol. Remember, we discussed it in our first 2 slides. The unabsorbed portion is acted upon by intestinal bacteria to form cholestanol and caprostanol. So what we got? We got the unabsorbed cholesterol. The intestinal microbiota, which is inside the intestine, they act on this cholesterol and convert it to cholestanol and caprostanol. These are excreted in the feces basically as a fecal sterols. Another 500 milligram of cholesterol is converted to bile acids, which are excreted in the bile as a bile salt. So this is how the excretion of cholesterol happens. So somebody in the Viva last time, if somebody asks you like, what is the amount of cholesterol in the feces? Please remember, we cannot say directly it's the cholesterol. You see the cholestanol and caprostanol. It's not directly the cholesterol as an answer. Okay, so remember this part, this tricky question sometimes. So you have to focus very clearly like what actually the mechanism is. So now what we got here, we got the liver cholesterol pool, and this cholesterol pool will basically give you the excretion of VLDL, free cholesterol, in the bile, and the conversion to bile into acids. And this is how the cholesterol is coming from up there, which is coming from the diet. So this is just showing the overall mechanism which we have discussed just now. Okay, now something very important. Sometimes it has been asked, which is about the role of liver in cholesterol metabolism. The liver has a major role in controlling the plasma level of LDL cholesterol. Number one, liver synthesizes cholesterol. Number two, liver removes cholesterol from LP remnants. Number three, liver is the only organ that can excrete cholesterol through bile. Number four, liver converts cholesterol to bile acid. So if somebody asks you this question, please focus on these a few points. Okay, now conditions with hypercholesteremia. What is the condition which okay, now something very interesting and very important to understand. We are talking about the cholesterol, but the issue is looking at our learning outcome. I focuses on hypercholesteremia. I did not focus on hypocholesteremia. Hypocholesteremia is another very important issue. So in the next coming lectures, I think in next coming modules, you're also going to cover that. But looking at the current circumstances, I focus on hypercholesteremia. So few diseases in which hypercholesteremia occur is, for example, diabetes mellitus is there. We have the obstructive jaundice is there. Hypothyroidism is there. Nephrotic syndrome and the familial hyperlipoproteinemias. It could also be like it could be genetic also. So these are some of the reasons why the cholesterol is high. For example, hypothyroidism, the receptor for HDL or liver cells are decreased, and so excretion is not affected. So if you look into the mechanism, all these mechanisms follow the basic biosynthesis of the cholesterol. Number two, obstructive jaundice, the excretion of cholesterol through bile is blocked. When the excretion is blocked, for sure the cholesterol become high inside the liver and for sure inside the blood later. In diabetes mellitus, acetyl-CoA pool is increased and more molecules are channeled to cholesterol. Nephrotic syndrome, albumin is lost through urine. Globulins are increased as a compensatory mechanism. So when they are increased, that is the lipoproteins is increased, cholesterol portion will be increasing with the lipoproteins because one of the constituents. And then hyperlipoproteinemia is the whole different topic, lot of genes, lot of molecules are involved. We are not going to cover, but at least you must know there is a term familial hyperlipoproteinemias. Okay, now this is something very, very important for all of you for the clinical years. That is hypolipidemic drugs. So the first one is HMG-CoA reductase inhibitors, and the other name for this one is statins, basically. Most popular treatment for cholesterol, high cholesterol is statins. So we do have atorvastatin and we have the simvastatins, which are popular drug in this group. They are effective in reducing the cholesterol level and decreasing the incidence of basically CAD. So secondly, we have the bile acid binding resins. Uh, for example, we have the cholestyramine and cholestyramine, which decrease the reabsorption of bile acids, and thus it affect actually the cholesterol excretion basically. So third one is Probucol increases LDL catabolism and prevent accumulation of LDL in arterial walls. So more cholesterol will be converted to bile acid, thus reducing the cholesterol level. Remember, this LDL has a role in the bile basically. So we have HMG-CoA reductase inhibitors, we have the bile acid binding resins, we have the Probucol. Okay, the reason I put all of them together is if you ever get a question on that, you know, you have everything compiled here, so easy for you. So you'll be also going through in different classes further on this actually. So remember, we have the HMG-CoA reductase inhibitors, we have the bile acid binding resins, and we have Probucol. Okay, now when to start the statins? Example, you have family members, you have your friends. They said, "Oh, I think my cholesterol is very high. I'm having a symptoms, you know, kind of in which you can feel. Example, there is an issue with the vision. There could be issue with different symptoms anyway. So what we focus now is the person come to you and they said like you know they feel they have high cholesterol, and when they check their cholesterol, in the lab the cholesterol is really very high. So what do you have to focus now? So remember these some points. Number one, all patients with established cardiovascular disease. So this is a secondary prevention. Number two, patients with diabetes mellitus, especially over 40 years of age, can start the statins. Number three, diabetes mellitus with other risk factors such as retinopathy, nephropathy, microalbuminuria, and hypertension should go for statin. Number four, total cholesterol above 220 milligram per DL or LDL more than 160 milligram DL or total cholesterol, that is LDL ratio more than 6, should go for statins. Number five, familial history of premature cardiovascular disease. Number 6, lifestyle alteration. Example, exercise and diet are not enough to reduce the cholesterol level. You know, sometimes you have the high cholesterol, and your friends and your doctor told you, please go for a jogging, go for exercise, but you feel it's too high, and maybe it's take 8 to 10 months to reduce it with the exercise, with the jogging. So then doctors normally suggest you to go for statins. So these are different expected effective drugs used for basically lowering the cholesterol: statins, fibrate, niacin, cholestyramine, and estimate. So you can go through just a table. It's not important for you. Actually, this slide is not important for you for this lecture, but just to add in a little knowledge to all of you. Okay, that's all. Thank you so much. Have a nice day. That is all lecture for today. So I try to make it very simple. Plus, I try my best. Basically, I hope you will not mind. I'm very clear. So I'm basically using different books. I'm copy pasting sometimes from the books for just for a reason you at least read everything. You know, otherwise making a lecture, because normally we have a habit, we normally go through the lecture these days, you know, we don't go for the books. So my whole idea is like at least you go through and you read everything. So that's why lecture becomes sometimes lengthy, but still, you know, you can go through, maybe useful for you in your class, inshallah. So that is all as per my promise. I think for the first time I finish early. Otherwise, normally I take more time. Otherwise I have to take after 11. Any questions you have, please, you are welcome. If you guys have any questions, you are welcome. If no questions, or frankly speaking, we are done with our lecture. Anybody would like to leave the class can leave. I will be uploading my lecture notes in Caleb. Plus, I'm going to WhatsApp the lecture notes to Afik in few seconds. He can distribute among all of you also. In case we are not sometimes, you know, accessible to our Caleb system from the home these days, so I will also WhatsApp to Afik. That is all from me. Thank you so much. You guys can leave if no question. Inshallah, well, inshallah, we'll see and we'll meet again very soon. Thank you so much for being attentive. Assalamu alaikum.
Salam. Thank you, Prof.
Thank you, Prof.
Salam. Thank you, Prof.
Thank you, Prof.
Thank you, Prof.